Okay. Hello, everyone. Good afternoon. If you're listening from the UK, that is. Welcome to today's edition of the 3D Regulatory Genome, and it's a great pleasure for me to welcome Dr. Sarah Marzi here as today's speaker. Sarah has graduated with degrees in mathematics and psychology in the University of Freiburg, and then did her PhD in epigenetics at King's College London. From there, moved the other side of the river to complete a postdoc at Queen Mary in London, and received an independent research fellowship at the Imperial College London, and now is back at King's College London. I hope I got that right.
That's all right.
Okay. Okay. So covered a lot of London institutes. Yeah. And Sarah's lab combines experimental genomic and epigenomic techniques with innovative statistical and computational analysis to decipher gene regulatory and epigenetic mechanisms and environmental risk factors contributing to the earlier stages of neurodegenerative disease. And before I hand over to Sarah, quick housekeeping. This session is recorded, so you can also see it later. We'll stop the recording just before the questions. And talking of the questions, I would ask that you ask them at the end. So either well, the easiest is if you put it in the Q&A box and I read them out. Okay. And with that, I'll hand over to Sarah.
Perfect. Thank you very much for the lovely intro. Great. I'm starting on the hidden slide. Here we go. So basically, you've explained already really well what we do. We're an epigenetics group interested in neurodegenerative diseases and sort of figuring out what the earliest cellular gene regulatory mechanisms are that link the causal onset factors of disease with it. So if we think about neurodegenerative diseases, they're a class of diseases in which generally neurons die somewhere in your central nervous system, most often your brain, and the dementias will affect regions of your brain that are important for cognitive processing, memory, and these kinds of processes, which is why you get the associated cognitive symptoms. But you also have more motor-related diseases like motor neuron disease or Parkinson's, where neurons either in your motor cortex or even in your spinal cord might be affected, and therefore those processes start to fail. But overall, neurodegenerative diseases actually, beyond just neuronal cell death, share a number of cellular and pathological features, and the most common being pathological protein aggregation. So virtually every neurodegenerative disease is associated with some form of protein misfolding and aggregation, and this can happen either within the neurons themselves or between the neurons. So if you take Alzheimer's, for example, you have two proteins that misfold, amyloid, or amyloid is a fragment of a protein actually. Amyloid builds these plaques that accumulate between neurons, and the other protein is tau, which is normally involved in the neuronal cytoskeleton, and that forms tangles within the neurons. And we know that both of these protein aggregates are bad for the neurons. So if you take neurons and you expose them to these aggregates, that's generally neurotoxic for them. But we don't really know why the proteins start misfolding in the first place and why the normal repair mechanisms and degradation mechanisms that should prevent this from happening are not functioning properly. So there are a number of other things that happen in the cells and in the tissue around this. For example, inflammation, neuroinflammation is a common feature across all diseases. You'll often see damage in the DNA and sometimes in the RNA as well. Energy production and metabolism is often altered, and mitochondrial function, cytoskeletal abnormalities, and like I said, aberrant proteostasis, so the mechanisms that should break down some of these proteins aren't really functioning properly. And then also as a consequence, or might precede even the neuronal death, you get a misfunctioning of synapses, so the contact points where neurons communicate with each other. And this can go both ways. So often, for example, in Alzheimer's, in the early stages, you'll see and overfiring of synapses, whereas later on they lose their activity, the neurons die, but also the downstream neurons that are failing to receive input from these neurons then become more vulnerable. Anyway, this is all I wanted to say about what's happening in these cells and at the pathological level, but we don't really know why it's happening. And if you think about that in the really big picture, there are sort of three big factors that can influence your risk and for these pathological mechanisms and for the diseases. Number one being genetics, genetic variants associated with diseases, and quite often the diseases we study sort of segregate into more familial and common forms. So take Alzheimer's, for example, about 3% or so of cases have familial forms of Alzheimer's disease, where individuals would carry mutations in three well-known genes, APP, which is the amyloid precursor protein, and two presenilin genes. All three of these genes are involved in production of amyloid, and therefore we've learned much about amyloid through these familial forms of disease, but they make up just the minority of cases. So 97% of cases don't have these strong genetic familial risk factors and mutations. And there it's way more common. You have and complicated because you have common variants associated with disease, each with very small effect sizes, and they probably interact with environmental risk and other things you're exposed to over your life course. And so it's much harder to dissect what their actual effects are. I just mentioned environmental risk factors. There are some quite common ones across diseases. I don't know, air pollution has been mentioned recently a lot as a very common one, but pesticides and toxicants, for example, are generally quite bad for your neurons and can have risks on a number of different neurodegenerative diseases. And then the last factor, I guess, would be things that are maybe unchangeable, like aging. Certainly the aging process predisposes us to these diseases. And in fact, for most of them, they are very late age diseases. And the single biggest risk factor in developing them would be your age. So above the age of 90, for example, you have a 50% chance of having some Alzheimer's pathology in your brain. And that only goes up as you age more. So then, to understand what both the environmental factors and the genetic risk variants are doing, we are using epigenetic annotation and studying genome regulation at that scale, because we think we can use it for both functional genomics and for understanding environmental risk factors better. And when I talk about epigenetic regulation, or what we mostly do is sort of chromatin and enhancer-driven gene regulation. So we study the histone modifications, the chromatin accessibility, but the downstream RNA as well. We do infer sometimes 3D genome regulation a little bit, but in our lab, we haven't done so much of the actual chromatin conformation experiments ourselves. Just for context on these two plots, so on the right, this is the most recent genome-wide association study of Alzheimer's disease. And as I mentioned, overall, the genetic architecture of late onset Alzheimer's disease is one that's primarily characterized by common polygenic variants, so variants with very small effect sizes. And the one notable exception to that pattern is the gene ApoE, apolipoprotein E, that I'm sure most of you have heard about. And it occurs in three isoforms, ApoE2, 3, and 4. And this ApoE4 isoform increases your risk for Alzheimer's disease quite substantially. In fact, if you have two copies of that isoform, your risk is increased up to 12-fold compared to the E3, which is sort of the common isoform. This is actually protective, but quite a rare isoform. And then on the environmental side, this illustration is an example from Parkinson's disease, because actually that's a disease that has a great environmental contribution. So that's less heritable than some of the other neurodegenerative diseases. It's thought to be only about 20 to 30% heritable, meaning the other 70% are explained by non-genetic factors. And we did a review recently on environmental factors that have been reported in literature. So these are all the ones where we have epidemiological evidence in humans that they're associated with disease. And we showed that there is increasing, but still early days, evidence that these can drive epigenetic patterns, and maybe that the epigenetic mechanism is the way they mediate their effect, because a lot of these are very low-grade chronic exposures over many years. And so the question remains, why do they cause such a very specific syndrome and disease state, but only after decades of exposure, for example? And so the epigenome in that sense could serve as a cellular memory of these low-grade chronic exposures over time. It would be quite informative. So I already told you that genetic variants associated with Alzheimer's disease are common and polygenic. In fact, they're also primarily non-coding, so they don't tend to fall into coding regions of the genome. And it's been therefore challenging, again, to decipher what are they actually doing. We have shown over time that they're not just anywhere in the genome, but they're in fact enriched in regulatory elements of the genome. So this is a study profiling H3K27 acetyl, which marks active enhancers and promoters in the human postmortem entorhinal cortex. So this is a region that's close to the hippocampus and that's affected quite early in Alzheimer's disease. And we showed, if you look genome-wide at the whole heritability and genetic risk for AD using a method called linkage disequilibrium score regression, you can show that the variants that fall into regions of H3K27 acetyl carry proportionally more of the heritability than those variants that don't fall into those regions. And in fact, that's actually true for a number of other neurological and brain-related disorders. So if you want to understand the genetic risk there and the mechanisms, you really need to study them in the brain. This was performed in bulk tissue, but since then there have been a number of really nice studies and with very consistent results that have pinpointed the genetic risk of AD to be primarily enriched in microglia. And this is by proxy and vicinity-based gene expression data here in the mouse using single cell studies, so showing that the genetic variants are enriched near genes that are specifically expressed in microglia. This is from Nathan Skeen, who's a colleague at the UKDRI. And then here, this is an example from another UKDRI colleague, Lexi Knott. And he purified different cell types from human brain resected tissue, including microglia, neurons, oligodendrocytes, and astrocytes. And he showed, again, that the genetic risk for Alzheimer's disease is microglia. And in fact, he showed that much more strongly so in microglial enhancers than in the promoters. So that tells us, if we want to understand what these genetic variants are doing and what context they're active in, what genes they're regulating, and what processes they're driving, we need to be studying microglia. And that was actually quite surprising, because for many years people had been focusing almost exclusively on the neurons, because they are the cells that form protein aggregates, they are the cells that die and misfunction. And so people had assumed that it must be the neurons that are driving disease, but it turns out actually the genetics is very consistently pointing to the microglia. And I should mention microglia are the brain's immune cells. They're a type of macrophage, so they're involved in a plethora of different functions, some of them being classic macrophage, so cleaning up, eating up debris, eating up anything that's dangerous for the brain. And so, for example, aggregates that are forming partly could be digested by microglia as well. So more generally, it's really hard to link loci to function, and it's even hard to link epigenetic loci to function. And one of our projects, side projects, so to say, to dive a bit deeper into this and to leverage some of our machine learning interests and expertise, has been looking at chromatin deep learning models. So these are models that are fed in epigenetic information, like information from histone modifications, and then try to predict gene expression based on very large windows upstream of the genes. And so this, I worked together with a former PhD student at Imperial, Alan Murphy, on this project, and we looked at ENCODE data because, in fact, to build or to train these chromatin deep learning models, we needed both the histone modification data, but also matched chromatin conformation Hi-C data so that we could link regulatory elements to their target genes. And so we had 7 histone marks plus the Hi-C data across 11 cell types from ENCODE. And we trained 2 different architectures, one quite a simple promoter architecture across the modifications, and the distal model that took 40 kilobase input and the Hi-C chromatin conformation data on top of that. And we wanted to see, I mean, this has been done successfully as compound models before, but we wanted to see which histone modifications predict best in which context. What if I wanted to profile the most informative for gene regulation, which histone modification would I choose, and those kinds of questions. And so we trained the model, and it works quite well, and it shows you some expected things. So I think the 2 histone acetylations, the K27 acetylation and the K9 acetyl, generally predict quite well, as does the H3K4 monomethyl. But it really also depends on context. And for example, stem cells have quite different patterns to mature differentiated cells. But the nice thing is, once you've trained the model, you can actually start to play with it, and you can perform in silico experiments and perturbation. So basically, where you have a histone modification signal, you can remove it or you can add it where there had been none. And you can see how the prediction of the model changes. And this is one example in a region upstream of the gene destrin. And this is the, in this case, it's a histone acetylation peak. You see the peak right here. And this peak actually contains a UK Biobank fine-mapped eQTL SNP for the gene destrin. And before we modify the region, you can basically look at attention or like what information is the model using to make the prediction of the expression here. And you can see before you perturb it, it's really not paying attention to this region at all. But if you take that peak away, it becomes really informative. And then the model is really just looking here. So meaning the histone acetylation signal at that point is really important for the expression of this downstream gene. And it's interesting because this is functionally potentially quite a relevant gene and could have applications for linking genetics to disease outcomes. So destrin is linked quite strongly to malignancy and lung carcinoma. And in fact, this was in a lung cell line, lung tissue cell line, I believe, or a cancer, lung cancer line. And so it's been shown that if you have high expression of destrin, you have much poorer progression. And in fact, also experimentally in mice, if you knock the destrin down, you get reduced or almost no metastases at all. Whereas if you overexpress, you make it much worse. So that's just to say one case in point. You can use these kinds of really in silico pure computational models and potentially infer interesting regulatory mechanisms that are disease relevant. And so going from the one interesting locus, we asked overall across the genome, what about the regions of the genome that are most predictive of gene expression? Are they interesting in other ways as well? And so number one, we showed that the regions where the histone modification signal is most predictive of the expression are also enriched in eQTLs. So that makes sense. The regions that have genetic variants that modulate expression probably act through epigenetic regulatory elements that sit on top of them. And then finally, we also asked whether they're specifically enriched in any genetic risk for diseases. And we looked, so here the limitation of ENCODE becomes very clear in that you're limited to the cell types that they have, and they're probably not cell types that are relevant to most of the diseases that we're interested in. So we specifically for this analysis looked at liver, which was one of the samples, and a few 2 liver-related diseases, and then neural progenitors, which was the closest we could get to brain, which is of course our organ of interest, and then a number of different brain disorders. And then we did a pan cell type most predictive marker as well. And as you see, only one of these enrichments is actually significant. This is the one for bipolar disorder, although NAFLD, which is a liver disease, sort of is the second closest that would come to more significant in the liver tissue. So I reckon if we could get these kinds of annotations and fit this kind of model in the right cell types, we could predict such things much better. And I would expect to see disease enrichment in the regions that are functionally most relevant. Yeah, and this was published just 2 months ago now in Nucleic Acids Research. So if you want to read more about it, it's all out there. But let me take you back to actual diseases that we're interested in. So one of my big first studies in the field is now several years ago, 2018. I profiled H3K27 acetyl in the human postmortem brain in Alzheimer's cases and controls. And I showed that you have really widespread dysregulation of the histone acetylation. So we're talking about thousands of regions in the genome showing differential acetylation between the cases and the controls. However, I just spent the first 15 minutes or so telling you how important the microglia are. And this whole study at the time was in bulk brain tissue. So another thing that we've been working on recently, in the last few years in my lab, has been computational approaches to deconvolute cell type specific signatures in bulk brain acetylation studies. This works surprisingly well because acetylation is very cell type specific, and that of course comes from the fact that enhancers are quite highly cell type specific. The work was led in my lab by Kitty Murphy, who's now a postdoc in my lab, and a bioinformatician at the time, Yuki Ye. They developed two complementary approaches to infer cell type proportions in a bulk tissue sample. So one based basically on a scar that sums average signal intensities across all regions that are specific to a given cell type, and one slightly more complex based on non-negative matrix factorization. So this is what is often used, for example, when you do cell type proportion inferments or deconvolution in gene expression data, and then you need to take care of a few other factors that are different in histone modifications versus when you're dealing with gene expressions, but it works pretty well and it's adaptable. And so we applied this to my original Alzheimer's data, and this is the matrix factorization approach. As you can see, it'll estimate the proportions of these 4 reference cell types, and the references are based from a published data set, in fact, from Alexei Nott's study that had profiles for each of these 4 cell types. And so for each sample, you get a proportion, and you can already see this visually by eye, but this is also quantifiable statistically and significant. The blue bars are bigger on the right than they are on the left. These are the neuronal proportions, and that is exactly what you would expect in an Alzheimer's sample, so that you get the loss of neurons as a main symptom or as a main characteristic of the disease. We did some validations with pseudobulk samples where we sampled together reads of known proportions, so mixtures of cell types, and showed that both of our algorithms can very highly accurately estimate the correct proportions on these. And then, as the most important thing, of course, we wanted to know, once we correct for those differences in cell proportion, like the loss of neurons that you have in Alzheimer's disease, where do the biggest changes epigenetically happen in the Alzheimer's disease brain? And so when we did this analysis, we did see some changes in the microglia, which is consistent with what the genetics would say, but also actually, and a lot more in both directions, downregulated and upregulated in the oligodendrocytes. And this was at first surprising, but there have been a few studies since that have been published on purified cell types in AD samples, as well as inferring gene regulatory networks that are cell type specific in AD mouse models. And all of these studies seem to consistently point to, when you look at the gene regulation and the epigenetics in an AD or AD model brain, the biggest changes are actually in the oligodendrocyte. And so some of these could be consequences of disease. They could still be disease modulating, or they might indicate that oligodendrocytes are potentially just a very plastic and responsive cell type in general. But this is something we're very interested in. It's a cell type that does, in fact, show functional changes quite early in AD, but that has historically not been studied very much at all. And it's something we're working on a bit further. And so just to say, this preprint was finally accepted for publication this week, just 2 days ago. So we're very excited about that. So the next study I want to take you into goes from looking at the whole genome-wide AD risk into one gene more specifically, and that's ApoE, the gene that I already mentioned. And this is a very relevant gene because the effects are so strong. In fact, it's not a familial variant, but some people have suggested if you have 2 copies of the ApoE4 gene, it's because it increases your risk so heavily. You can almost consider it a familial variant. And we were specifically interested in studying ApoE in microglia. Number one, because hopefully I've convinced you that the majority of general AD risk affects microglia. And microglia have been studied quite a lot in the last 10 years or so and characterized with single cell expression profiling to reveal a plethora of different activation states. And one that has come out as really interesting from this seminal paper from Ido Amit's lab is the so-called disease-associated microglia phenotype. These are microglia that are sort of highly activated, partially some inflammatory responses are upregulated, and they tend to localize just around the amyloid plaques in the Alzheimer's disease models and also in human Alzheimer's disease brains. And when you look at what characterizes this state, you can actually see that the gene that most upregulates its expression is ApoE. So it's heavily associated with this disease-associated microglia state. And then we also know that ApoE is in fact expressed by microglia and upregulated in these AD microglia. So microglia is a good place to understand ApoE and to understand the cell intrinsic function on the microglia. And so to do this, we paired up with Bart de Stroeper and Renzo Mancuso, both of whom work at the VIB in Belgium, and Bart also at the DRI here in London at UCL. And they, several years ago, developed a super elegant and I think quite sci-fi model of xenotransplantation, where they can take human microglia, for example, derived from iPSCs, and they xenotransplant them into newborn mouse brains. So first they enzymatically deplete the mouse's own microglia, and they inject the human microglia, and they spread out quite nicely across the mouse brain. When you look at the general homeostatic markers, they express all the ones that microglia should be expressing. They spread out well in terms of their nearest neighbor distance and distribution, and they integrate just as the mouse microglia would in a non-depleted mouse. However, they still look very much like human microglia in the sense that they're larger and have a more complex morphology. So we used this very nice system to specifically study the effects of ApoE variants on microglial regulation in a neuropathological context. So we want a mouse that actually builds these amyloid beta plaques because they're relevant for eliciting microglial responses that are disease relevant. And so we took an isogenic ApoE line with ApoE2, 3, 4 genotype and also an ApoE knockout, and xenotransplanted that into an Alzheimer's mouse model. This is called the APP-NLGF mouse. Basically, it has 3 human mutations and it leads to amyloid aggregation over the course of several months. So this was transplanted 4 days after birth, and then we wait 12 months. At that stage, they should have very extensive amyloid pathology in the brain. You harvest the brain, you can purify the human microglia based on human-specific cell surface markers, and then we performed a TAC-seq for open chromatin and RNA-seq for expression. And all the experimental work in our lab was done by my then postdoc Dihu, and all the analyses by Kitty Murphy. And so we see a whole bunch of different transcriptomic and epigenomic or chromatin accessibility changes in these microglia. One gene that struck us quite strongly because of the size of the effect and the consistency of the effect was CHCHD2. This gene is it's a mitochondrial protein, but again has such a variety of functions that it's hard to pinpoint what exactly it's doing here. For example, in response to hypoxia, it can become a transcription factor and localize to the nucleus and regulate things there, but it's also involved in cell motility and potentially chemotaxis. It's interesting because it has some rare mutations in the gene that are associated with Parkinson's disease, so a different neurodegenerative disease. And what we see is that it's very heavily downregulated here in terms of the expression in the ApoE4 variant. Here again, if you look across all the 4 cell types, this is the expression. You see that it's virtually not expressed at all in the E4 and actually not in the knockout either. And very concurrently with that, we see the same thing at the chromatin accessibility level. So this is the gene transcribed right to left. And you'll see these nice promoter peaks in the E2 and in the E3 genotype, but virtually no chromatin accessibility signature at all in the E4 in the knockout, and no expression either. That's just one gene that showed quite a striking result, but obviously we don't normally go one gene at a time. We want to ask what's happening a bit more systematically overall. So to do that, we use gene co-expression network analyses, WGCNA. And in fact, one of the modules that most strongly associated with the different ApoE genotypes, so you can see high expression of this module in E2, medium in E3, low in E4. When you look at what genes are in this module and what pathways and processes do they associate with, it's very much a cell proliferation and cell migration module, including processes like chemokine production and chemotaxis, so migration under immune activation, and also sort of in linking back to the CHCHD2 gene there. So proliferation and migration are standard sort of functional functions that microglia should have in their immune role. And we wondered if there are other processes of related or downstream of that motility that would also be affected, like the phagocytosis. So we got a list of known phagocytosis genes that are upregulated in phagocytotic microglia, and in fact showed that so this is now reversed, but they're basically enhanced in the E2, but the picture is the same. So cell motility is up in the E2 and cell motility is down in the E4, and the same is true for phagocytosis, where we see enhanced phagocytosis genes in the E2, and much less so in the E4, probably less than in the E3. We were also able to look at this more functionally in vitro with the same series of ApoE microglia, staining them for E. coli particles and myelin. So these are two things that they should be phagocytosing. It's slightly different processes, but both things that should be consumed by microglia. And when we quantified this, we showed, in fact, some significant differences across the genotypes. So number one, you can see that the E. coli particles, the ApoE2 is much better at phagocytosing them in the sense of each microglia takes up more E. coli particles. Whereas if you look at the myelin, it's the other way around. So the ApoE4 just has fewer microglia that are actually taking up the myelin fibrils. And there's a really strange thing also going on with the knockout that seems to take up a whole lot of myelin. So myelin is mainly like fatty lipids and fats. And so we think that's actually a toxic process where it's not able to process them and break them down. And so it's basically filling itself up with this myelin. But we haven't investigated that any further. So here we have enhanced motility and proliferation, and also enhanced phagocytosis in the E2. What else have we found? So there were some other modules that we saw dysregulated, and one of them was immune response. And so here in this immune response module, we see that it's enhanced in the E2 and down in the E4. But what exactly does it actually mean in terms of cytokines and immune signaling modules? Actually, pro-inflammatory cytokines are so basically upregulated in the E4 and down in the E2 generally. So these are 4 cytokines, and you see the first 3, they all have this trend where they have higher expression in the E4 compared to the other genotypes. This one is reversed, and this one is quite interesting because in the periphery and peripheral macrophages, it's known to be pro-inflammatory, but actually in certain tumor context in microglia in the brain, it's been shown to downregulate the pro-inflammatory response and have more anti-inflammatory properties. So what we think is the E2 has a more beneficial immune response, and the E4 has this high upregulation of pro-inflammatory cytokines. And in fact, that's been shown in 80 mouse models by the lab of Alison Goat a few years ago. And that's only been shown for the E4, but basically we also show that the E2 here again seems to be better at these processes than the other genotypes, and might have some of its protective effects through more anti-inflammatory action. And so when we wanted to understand we talked a lot about the gene expression landscape when we wanted to understand what might be the upstream drivers of these gene expression responses. We did some transcription factor binding motif enrichment analyses. And so this is in the regions that have increased open chromatin in ApoE2. And the top motif that we saw here was the vitamin D receptor. And so this was really, really interesting. Vitamin D receptor, as the name suggests, is activated by vitamin D. It's a nuclear transcription factor which has to heterodimerize with the retinoid X receptor and then binds in vitamin D response elements in the genome. And in fact, is known to have a bunch of immune regulatory functions. But it's also interesting because vitamin D has been linked to neurodegenerative diseases and specifically Alzheimer's, where vitamin D deficiency increases both your risk of developing Alzheimer's, but also makes your progression worse once you have Alzheimer's. So if you are vitamin D deficient, you will progress faster through disease. So we were kind of really interested to see this transcription factor come up. That was at the chromatin accessibility level. And we also wanted to check, are the vitamin D response genes then also upregulated in the E2? And in fact, we got a list of vitamin D receptor target genes. This was from monocytes, which was the closest cell type we could get to in published literature. But in fact, those response genes are actually upregulated in E2 when compared to E3. And here, this is formulated the other way around, but down in E4 versus E2, meaning up in E2 versus E4. So enhanced expression of the target genes. And so what we think is happening, one famous target gene of a vitamin D receptor is IL-10 and the IL-10 receptor. And so in fact, we see increased expression of the alpha subunit of the IL-10 receptor in the E2 genotype. And so overall, what we think is happening is that some of the protective effect of the E2 genotype might be conferred through anti-inflammatory action that is mainly driven, or partially at least driven, through the enhanced action of vitamin D receptor. And so that'll activate IL-10 receptor. And downstream of that sits SOX3, which is known to downregulate a whole cascade of pro-inflammatory cytokines. And so this work is out as a preprint for now, hopefully soon as a full paper. So again, you can read up more on this online. So just to summarize what I've been telling you a whole lot of stuff, but number one, the important point is non-coding genetic and environmental risk can act on these complex neurodegenerative diseases through gene regulatory mechanisms. And specifically, what we've been able to learn broadly about the genetics of Alzheimer's disease is that they're enriched in these microglia specific enhancers. And so if we want to understand their function, that's where we need to look and what we need to study. In the late AD brain, if you look at epigenetic regulation, you see both microglia, but also oligodendrocytes heavily dysregulated at the epigenetic level. So that's in contrast to the genetic enrichment. And then in the last part, I showed you that ApoE actually induces different regulatory responses on neuropathology-associated microglia. What I didn't show you is that it actually overlaps. So the genes that are regulated by ApoE4 overlap with the late onset AD GWAS risk genes. So that was quite interesting as well. But what I did show you is that you have this exacerbated cytokine response in E4 or vice versa, anti-inflammatory signaling in the E2. And that might be in part mediated by enhanced vitamin D receptor binding in the E2 genotype. So in the last 15 minutes or so, I wanted to shift focus and talk a bit more about the environmental side of our program, which primarily for now has focused on Parkinson's disease, because Parkinson's has this stronger environmental impact. And in fact, beyond just these associations and a hypothesis, or for some patterns like cigarette smoking, there are also some closer links between these factors and Parkinson's disease and the epigenome. So for example, several pesticides, including dieldrin and rotenone, are known to act directly on histone acetylation, and paraquat here, it can interact with alpha-synuclein, which is the protein that aggregates intracellularly in Parkinson's. or, for example, alpha-synuclein as a consequence can sequester DNA methyltransferases outside of the nucleus. So if you have neurons that are affected by alpha-synuclein aggregates, you would expect to see consequential epigenetic changes. And to begin with, and to pick one form of exposure, we actually started focusing on pesticides because there are very strong epidemiological links in human studies, and also some of these pesticides can be modeled quite well in animal models and in vitro. And so this was work led by my, at the time, PhD student, who's since left the lab, Maria Zalanchuk. And we picked the pesticide rotenone, which is a mitochondrial complex 1 inhibitor, which is fortunately not used in these regions of the world anymore, although there are still wide parts of the developing world, if you will, where like India, Africa, Brazil, where it's still used quite heavily as a pesticide. And to model it at different scales of temporality, complexity, cellular complexity, and organismal complexity, we chose this sort of this all-around approach. So we worked in the simplest model with a dopaminergic cell line to model the dopaminergic neurons that are the ones that degenerate in Parkinson's. Here we have very good control over the dosage, the temporality, the time course. It's relatively, like we can easily do lots of experiments, and so we can generate a lot of assays, and we just have one cell type, so we stick with bulk methods. The most complex system is probably this rotenone rat model, which our collaborators in Pittsburgh ran. And so here rats are injected with rotenone, and we can look at their brains afterwards. And we did some bulk analyses in ChIP-seq, and we're also doing some single cell expression and attack-seq profiling in that model as well. And the midbrain organoids are in theory a really nice in-between system where you have human cells, you have multicellularity and complexity. Our first pilot, we've piloted this, but it's not been all that successful yet, so that's more for the future a little bit. But I want to tell you primarily about the rat model, because I think it's a very nice and complex model, and we had some really cool first insights into what happens in response to rotenone in the rats. So this is the model. The rats, these are aged wild-type Lewis rats. There's no genetic manipulation or anything. They are injected over a time course of 21 days with low doses of rotenone intraperitoneally. And at the end of this, they're sacrificed, and we're primarily interested in a region of the brain called the substantia nigra. This is a region that is highly enriched in dopaminergic neurons, and this is the region that degenerates in Parkinson's disease. So it's basically primarily or almost exclusively the dopaminergic neurons in your brain that die in Parkinson's. And that's why, because this region has a really high density of dopaminergic neurons, you get a big loss of neurons in that region. We also actually sampled as a second area of interest the motor cortex, which is sort of downstream affected by projections from the nigra, and also, of course, involved in controlling movement and motor function. And so one of the cool things about this study is that we show actually region-specific differences in the response to rotenone. But just to tell you, these are our rats, so if you inject them with rotenone over time, actually they experience weight loss over the 21 days, the rats that get the rotenone versus the controls. And also you can quantify nigrostriatal degeneration of dopaminergic neurons, and you do this by proxy by staining for tyrosine hydroxylase, which is produced by the dopaminergic neurons, and then you see a marked reduction of this in the rotenone exposed animals. And this has been a long-standing and fruitful collaboration with Tim Greenemeyer and Emily Rocha in Pittsburgh. So Tim first developed this model in the early 2000s and has been researching pesticides and Parkinson's for a long time. Okay, so when we look at the epigenetic dysregulation, this is H3K27 acetyl ChIP-seq, in both of the regions, we actually see more significant loci in the cortex, but the most strongly significant changes are seen in the nigra overall. So number one, what do we see in the big picture? And actually, this is something that's reflected in both of the brain regions. We see very common stress and reactive oxygen responses that we would expect as a direct consequence of the NIGRA. So here is the association for the chip and the matched RNA as well. So for example, we see all these apoptotic pathways, responses to various stimuli like hydrogen peroxide, reactive oxygen species, response to hypoxia here in the cortex as well, or peptide hormone stimulus. And the second thing that's quite in common here was this actin filament organization. So this potentially involving the cytoskeleton and maybe also related to the reactive oxygen species and DNA damage response. And that was the, yes. And so we didn't, again, this was just based on the overall dysregulated genes, but we also performed gene regulatory network analysis again, so these co-expression networks, and again the modules that are changed and that are consistent. So you see a response on cellular respiration, or here aerobic respiration in the nigra and in the cortex, which we see down-regulated in both, or also here mitochondrial complex activity. And this is exactly the stuff that rotenone directly affects, right? It's a complex 1 inhibitor. So yes, it will inhibit mitochondrial activity, and it will inhibit cellular respiration, aerobic respiration. And so these are the strong gene expression responses that you see in the exposure to rotenone. What we were a little bit more interested in was what distinguishes the responses in the nigra to the cortex, because the degeneration really just happens in the nigra, and why is the nigra so specifically vulnerable to it, and what happens in the nigra. And so number one, what we saw when we looked at regions that were specifically only changed or here upregulated, hyperacetylated in the nigra, we see the strong annotation and enrichment of really immune pathways, so including chemotaxis, activation of various types of immune cells, including and proliferation of T cells, granulocytes, neutrophils. And this seems to be relatively specific. So this is just based on regions that are specifically up in the nigra and not in the motor cortex. And when we do the motif analysis of this and look for enriched motif, actually we were struck that virtually all of the enriched motifs that we observe are immune-related motifs. So that includes the STAT family, for example, or the IRF family. These are interferon regulatory factors, so they're upstream of bigger interferon gene expression and responses, or also the Jun-Fos complex. And we see upregulation of some of these both at the RNA, the chip level. So both the motif is enriched in the chip, hyperacetylated regions, but also the ones marked here with the star, there we also see an increased expression of that transcription factor itself. And so these are our top candidates of what might be driving these responses. We were also quite interested because its role has been discussed more and more recently in the complement system, and its role in microglia, in particular in Alzheimer's, has been discussed along, and we have collaborators in Cardiff who've been working on it for decades. So they gave us their curated list of complement genes to look at, and we showed in fact that the complement genes are actually upregulated both in the cortex and in the nigra, but more strongly so in the nigra. But we were really struck to see that it's complement component 1, C1q in particular, that shows both extreme upregulation of expression and acetylation in the nigra. So here we show both of the brain regions, and C1q is built of these 3 C1q chains, so the alpha, beta, and or ABC chains. And then forms the C1q complex together. And we see that all 3 of them are massively upregulated in nigra. They're already more expressed in the nigra, and that might be due to the higher proportion of microglia in the nigra, but the upregulation is really quite extreme and also specific to the nigra. And then when you look across the genomic region, because the 3 genes are basically encoded one right after the other, the annotation is missing, but these are the 3, CA1Q, C1Q, B, and C, and we see across the whole region in the nigra we have this increased histone acetylation signature that's not present in the vehicle as well. So very specific specific, regional specific immune upregulation, particularly of the complement system and cascade. And C1q, I should say, is very much at the very beginning of the classical complement cascade. So it should be an activator of that whole process. Conversely, when we looked at the motor cortex and you look at the pathways that are enriched and that are unique to the cortex, they're really processes related to neurons and synaptic activity and organization. And here the interesting thing is that it doesn't seem to be just going in one direction. It's not a loss of synapses or a gain of synapses. Some of them are upregulated, some of them are downregulated, some of them in both directions. So both in the hyper and hypoacetylated region. So it just seems to be an overall dysregulation or loss of control of regulation of synapses. One gene that struck us in particular from our analysis, because it's a known upstream regulator of various other synaptic processes, is HOMR1. And so this one is actually upregulated in the rotenone condition, and all the downstream genes or interactors of HOMR1 are consistent with this, are very strongly upregulated as well in the cortex, but not in the nigra in our rotenone samples. So overall, what we see is these, we see some very strong classic responses to the exposure that relate to mitochondrial function, respiration, and reactive oxygen species that are very consistent across the brain. And I would expect quite consistent probably across any tissue in your body, because that's the primary function of the rotenone. But then we also see these really interesting, unique signatures in the nigra of this immune upregulation that could, in theory, take place in the cortex as well, but doesn't. Like the same cells are present, so that it could mount the same response, but it doesn't. And we see this unique synaptic dysregulation in the cortex. And again, we've been working on all of these projects for several years, and the last few months have just been the period where we have the big preprint and paper dump. So we put out the preprint for this in December. So again, you can go in and read that in more detail. Oh, I'm running out of time, I can see. So I just summarized that already. I'll skip this. And just to tell you in 2 minutes that we've been working really hard on doing single cell work on these samples as well, because of course we want to learn much more specifically what's going on in individual cell types. And that's been a whole odyssey, because it's actually extremely hard to perform these single cell experiments on the very small rat substantia nigra, which starts with 2 milligrams of tissue. So this is our cascade of optimizations. It took about 2.5 years. And now we have a beautiful data set from 7 control animals and 7 rotenone exposed animals with over 115,000 nuclei, all generated by Maria, and my current PhD student Paulina is analyzing all this data. We have all the cell types that we expect or would be interested in it, like the dopaminergic neurons, astrocytes, microglia. And I'm not going to show you a lot of results, just one overall finding that we're seeing in response to rotenone, as we'd sort of seen at the very crude bulk level. We see, in fact, an immune glial activation following this rotenone exposure. And we see this in the proliferation and upregulation of 2 types of astrocytes. In our data for now, they're labeled astrocyte 1 and 2. But in fact, this astrocyte 2 basically corresponds to a state that was published in the literature by Shane Liddelow and termed reactive astrocytes by him. And we also see a similar upregulation of various microglia types, called in our data microglia 1 and 3, but actually they respond specifically to 2 states reported by Renzo Mancuso as cytokine response microglia and antigen presenting microglia. And these, again, both proliferate but also upregulate their expression in the rotenone condition. And there's a lot more to dissect in that data, but I'll tell you about that another time. So I won't take any more talking time. I'll just thank you all for listening. Thank all the members of my lab that I've mentioned throughout, and the collaborators and the alumni throughout that were involved in these projects, as well as our funders.
Thank you very much, Sarah. That was fantastic. Tour de force.